Okay, we're going to pick up where the last video cut out before moving to our major topic for today, which is filtration. Our last video cut out as we finished up our review of blood vessels, and then we were about to start looking at the processes that happen in the kidney. So let's quickly review the blood vessels. Blood comes in to a nephron by way of the afferent arteriole. And the afferent arteriole is being highlighted here. That afferent arteriole leads into a network of capillaries known as the glomerulus, and then blood that leaves by an efferent arteriole. Now that efferent arteriole is going to divide back up into a series of capillaries known as peritubular capillaries. The prefix peri means around, so these are around the tubule capillaries. And there's actually two different types of nephrons. There's ones that stay kind of towards the outer rim of the kidney, and those are called cortical nephrons. And then there's ones where this loop of Henle dips really far in towards the center of the kidney, and those are called juxtamedullary nephrons. And if you have a juxtamedullary nephron, this is one where the loop of Henle goes really far in. It's really the only one we're going to talk about today. Then the peritubular capillaries go by another name called vasa recta. I don't use the term vasa recta in my exam. I just use peritubular capillaries to describe all of them. But in anatomy, you may have learned about vasa recta, and I just wanted you to realize that that's a name given to the peritubular capillaries when they wrap around the loop of Henle, but only the loops of Henle that go all the way down into the medulla of the kidney. Okay, so now that I've said all that, you can pretty much scratch that from your brain, and we'll just use peritubular capillaries here. And this is an example of what is known as a portal system. So it's not terribly common to find portal systems in our body, but when we do, they always serve a purpose. And here is an example of a portal system. A portal system is where you go from an arteriole to a capillary to another arteriole to another capillary before finally getting to a venule. Remember, the traditional route of blood is to go from an artery to arterioles to capillaries to venules to vein. And here, it's almost like we're duplicating the arterial and capillary bit. Hopefully, the purpose of this portal system will become evident once we get into reabsorption and secretion. But for now, we just need to know that blood kind of follows this interesting path. Now, I want to bring up another term here called a renal corpuscle, which you may hear of. The renal corpuscle is a term that basically describes the glomerulus and Bowman's capsule together. So if I'm just referring to the capillary bed, that's the glomerulus. If I'm just referring to the nephron bulbous kind of head that envelopes the capillary bed, that's Bowman's capsule. If I'm referring to the entire structure, all of this together, that's going to be a renal corpuscle. Just want you to be aware of that term. It may show up in your test. There's another term that I want to introduce you to, and it's one that will become a bigger player later on in this lecture series, and that is something called the juxtaglomerular apparatus. It's actually a big term, so go ahead and pause the video and write the term down. And then know that we often use an abbreviation to denote the juxtaglomerular apparatus. The juxtaglomerular apparatus often gets abbreviated JGA. Now, the juxtaglomerular apparatus is such that when we actually take a look at a real nephron, it is folded not in the way that I drew it in the previous lecture where I kind of unfolded it and splayed it out for you, but rather it follows this kind of interesting path. And it follows this path for a reason. So let's go ahead and trace this path. I'm going to actually use this part of my drawing and trace it. And we're going to name the parts of the tubule as we go through it. And then we're going to take a look at what that juxtaglomerular apparatus is. So we start with Bowman's capsule. This right here is the proximal tubule. The proximal tubule dips down into the descending loop of Henle, and then the ascending loop of Henle. And where the ascending loop of Henle transitions to the distal tubule is right around where the afferent and the efferent arteriole are. In fact, if my fingers represent the afferent and the efferent arteriole, it's almost like the ascending loop of Henle kind of does this, right? So it actually kind of sits in there and forms this really interesting complex. And that is the juxtaglomerular apparatus. And just real quick, this is the distal tubule, and then it would feed into a collecting duct. So the juxtaglomerular apparatus is actually this complex right here, and it's blown up right here. So you can see the afferent arteriole, you can see the efferent arteriole, and you can see a cross-section of the tubule where the ascending loop of Henle transitions into the distal tubule. So let's go ahead and write up the three parts of the juxtaglomerular apparatus. Now, where that nephron is when it talks to the afferent and the efferent arteriole is really very close to the transition between the ascending loop of Henle and the distal tubule. And it's not always clear where that transition is. I tend to refer to that tubule as a distal tubule, but you may also see it referred to as the very end of the ascending loop of Henle, and it really is right around that juncture. Now, the word juxta, think like juxtaposition. So this is a juxtaposition, like an arrangement where you're bringing things together. Of what? Well, the glomerular apparatus. Well, where have you seen glomerular before? How about glomerulus? Right? So the glomerular apparatus. So this is actually a juxtaposition of the early distal tubule with the afferent arteriole and the efferent arteriole, and they all talk to each other. Now, the reason for this juxtaglomerular apparatus will be saved for another lecture, but I wanted to introduce you to the term and show you what it is because it will become important later. So now that we've finished our review of the anatomy of the nephron, let's take a look at some of the functions. Okay, there are multiple functions that are traditionally associated with the kidney, and they are filtration, secretion, reabsorption, and excretion, in no particular order. So what we're going to do is we're actually going to describe what these processes are, and then we're going to map them onto our nephron to show you where these things happen. Okay, let's describe filtration. Filtration is the process of taking the blood and kind of squeezing out the finer particulates and fluid. And the analogy I'm going to give you is actually from Finding Nemo. If you have seen the movie Finding Nemo, a major plot element is to have these fish that are trapped in a fish tank try to actually ruin the filtration system in that fish tank as a means to escape. And they do a pretty good job until the dentist actually replaces the filtration system. And the dentist replaces the filtration system with this like new super, I forget what it's called, but it's like some super amazing filtration system that basically sucks out some of the aquarium water and runs it through basically like a pad. And the pad is kind of bluish and has some kind of fuzzy, it almost looks like a sponge. And the idea is that the fluid is going to like run past that sponge, and all of the particulates are going to get trapped in that sponge, and then purified fluid goes back into the fish tank. And at any given time, maybe 5% of the fluid is being removed and filtered. And so if it's allowed to run continuously, all of the water continues to be purified, again, just by removing a little bit at a time. And the kidney kind of does the same thing. It takes some blood and it kind of squeezes out some of that the finer parts of the blood, which would be plasma and small particulates, while filtering out or keeping behind the bigger elements like the cells, red blood cells, white blood cells, and platelets, and then only allowing the fine stuff to go through. So that happens at the renal corpuscle. So we can put a big F here. That's not a grade. That represents filtration, to show you that the process of filtration actually occurs at the glomerulus and Bowman's capsule. So the idea is that blood is going to come in through the afferent arteriole, kind of get trapped a little bit in that glomerulus, and that some of the watery parts of that fluid are going to get squeezed out and then captured in this tubule. And then what's going to happen is the fluid, which is now called filtrate, so we actually have a name. Once the fluid has left the blood, it is called filtrate. That fluid is going to basically travel all along the nephron tubules. And while it's traveling along the nephron tubules, it is going to be exposed to interstitial fluid here and also blood vessels, right? These are the peritubular capillaries that are going to kind of be right next to it. So we'll put in peritubular capillaries. And so we have this exchange between the extracellular fluid and the filtrate. How much filtrate is produced per day? And the number is actually astounding. If I were to take all of your nephrons, and there's, probably over 1 million in your body, and do a cross section right here and kind of measure how much fluid crosses all of those spots in all of the nephrons per day, it would equal 180 liters per day. This is actually measured. It's not measured directly the way I said. It's measured through some proxies, but it has a term. It is called GFR, which stands for glomerular filtration rate, and it's something we're going to spend quite a bit of time talking about, so I'll introduce it here. So GFR is glomerular filtration rate. So I want you to think about what 180 liters per day looks like. I mean, how many liters of blood do you have? Not 180 liters. You have, not even close to that, right? So this is probably like 20 times the amount of volume in your body. So if you were to actually let all of that continue until it reached the collecting duct and then went down the toilet, literally, you would dehydrate yourself to death within probably about an hour or less. So obviously that can't happen or we're dead. So what we want is to actually put most of what we filter out back into the body. And so the body has all of these interesting ways, really interesting ways of kind of recapturing that stuff. And we have a name for it. It's called reabsorption. So the process of reabsorption is recapturing materials that are now in the filtrate and putting them back in the interstitial fluid. And from the interstitial fluid, they're going to go back into a peritubular capillary and get put back into circulation. So that includes almost all of our water. We want to keep most of our water. We also want to keep most of our salts. We want to keep our nutrients. And interestingly enough, we might even want to keep some of our waste products like CO2 or bicarbonate. So the idea is that a lot of this tubule is dedicated to this process of reabsorption. And this is going to be covered in a totally different set of lectures, but I wanted to introduce it to you now. So reabsorption, which I'll just use as an R, is going to happen in the proximal tubule. It happens in both parts of the loop of Henle. It happens a little bit in the distal tubule, and it happens in the collecting duct. So some type of reabsorption is happening pretty much throughout the length of the nephron. Now, what is secretion? Secretion is actually kind of the opposite idea. If reabsorption is the idea of taking what is in filtrate and putting it back into our body, then secretion is taking extra stuff that's from our body and dumping it in filtrate, usually with the express intent of having it be excreted as urine or as part of urine. So if we think of reabsorption as going this way, this is reabsorption, then secretion is actually the opposite. So some secretion happens in the proximal tubule, and some secretion happens in the distal tubule. And those are kind of the major spots. There's actually some secretion that happens here as well, but I don't talk about it. So I want you to know that secretion occurs in the proximal tubule and the distal tubule. Okay, so we start by filtering a huge amount of this fluid, and that becomes our filtrate, and then as it goes through the nephron, we pick out what we want, reabsorbing, secreting a little bit, but mostly reabsorbing. It's mostly reabsorbing that's happening as you go through that nephron. And then basically once you get down here towards the end of the collecting duct, where that collecting duct feeds into like a minor calyx, if you remember that from anatomy, then there's no more alterations happening to that filtrate. We're not doing reabsorption, we're not doing secretion, we're not doing anything else. And it's basically at that point excrement. It's urine. So eventually it will travel down a ureter into the bladder, and then from the bladder into the urethra, hopefully into a toilet, but maybe not. So at that point, we call that urine. And so the process of excretion is going to be down at the end of the collecting duct. So in our renal lectures, we're going to spend most of this series of lectures will be broken up into a couple parts, looking at filtration. Then we're going to spend a lot of time looking at reabsorption, and we'll look at a little bit of secretion. I could teach an entire class on renal physiology and not run out of material to teach. That's how much stuff is going on. But we're kind of picking our battles here and just giving an overview to how this works. Excretion is not something I really spend any more time on because, once you've made urine, it's just about getting the urine out of the body. Okay, so that's where we're going to stop on this lecture. In our next one, we are going to formally look at filtration, what it is, and what regulates it. I'll see you in the next lecture.